Welcome to this session. In this session, we are going to learn about neuromuscular junction. To begin with the introduction, skeletal muscles are innervated by motor nerves. So just see this diagram. So here you can see we have a muscle which is innervated by a motor nerve fiber. What is this motor nerve fiber? Motor nerve fiber is an axon of the motor neurons, where the anterior column of spinal cord contains the motor neurons. There are two different types of motor neurons. This is the alpha motor neuron. So the cell body of the alpha motor neuron lies in the anterior column of the spinal cord, whereas its axon is going to innervate the skeletal muscles. So a motor nerve fiber divides into number of branches, and each branch of the motor nerve fiber innervates muscle fiber. So there is a nerve terminal which is going to innervate the muscle fiber. So you know that is the motor nerve fiber, which is an axon of the spinal motor neuron. And what is the definition of neuromuscular junction? Neuromuscular junction is also known as myoneural junction. It is also known as myoneural junction, and it is the intimate contact of the nerve endings with the muscle fiber to which they innervate. So it is simply a junction between the nerve terminal and the muscle fiber. It is a form of chemical synapse because the nerve ending is going to release a neurotransmitter which is going to act on the muscle membrane, which is going to initiate the nerve action potential in the muscle, which is going to cause the muscle contraction. So it is a form of chemical synapse. We will study detailedly about when we study the synapse. There are different types of synapses, like physiologically we can classify synapse as chemical synapse and electrical synapse. In chemical synapse, some chemical will mediate the transmission of impulse, whereas in electrical synapse there will be gap junctions between the two neurons. So the nerve impulse will transmitted from one neuron to the other neuron through that gap junction. So that is a type of synapse which is called as electrical synapse. So neuromuscular junction is formed at the point of contact of spinal alpha motor neuron with the skeletal muscle fiber. So it is the junction between nerve terminal and the muscle fiber. Here you can see the structure of the neuromuscular junction. So when we study about the structure of neuromuscular junction, we should remember four important structure. The first one is synaptic knob or nerve terminal or terminal button. All these are same, which is also called as telodendria. And the second important structure is called presynaptic membrane, which is the membrane which is lining the axon terminal or nerve terminal. And then this is the postsynaptic membrane, which is nothing but the muscle fiber membrane. So you know, one side there is a nerve terminal, the other side there is a muscle, right? So this is the presynaptic membrane which is lining the axon terminal, whereas this is the postsynaptic membrane which is the muscle fiber membrane. In between this there is a space which is called as synaptic cleft. So these are the four important structures we should remember while studying the neuromuscular junction. So this is the synaptic knob or a terminal button or nerve terminal or telodendria. This is the presynaptic membrane, which is the axonal membrane. Then there is a synaptic cleft, which is a space between the pre and postsynaptic membrane. And the postsynaptic membrane, which is the muscle fiber membrane. Here you can see the axon. So already we have studied the structure of a neuron. So the axon, which is a long process of the neuron, it is lined by myelin sheath. So here you can see the axon loses its myelin sheath and divides into number of fine branches, and these nerve endings, they will end as small swellings. Those are called as terminal boutons or synaptic knob. So this synaptic knob, it contains synaptic vesicles, and these synaptic vesicles will contain the neurotransmitter, that is acetylcholine here. So this synaptic knob also contains mitochondria. From where this acetylcholine is synthesized and stored in this synaptic vesicles. So the acetylcholine is synthesized in the cell body of the spinal motor neuron. So we have seen in the previous picture that in the anterior column of spinal cord, it is the place where the alpha motor neuron, that is the cell body of the alpha motor neuron, lies in the anterior column of the spinal cord. So from the cell body, because cell body is the protein machinery, it is the one which contains the nisyl granules, ribosomes, everything. So it is the place where the neurotransmitter and synaptic vesicles are synthesized, and they are transported from the cell body to the axon terminal. And And that type of transport is called as fast axoplasmic transport. And we should also remember that there are more than 3 lakh synaptic vesicles are there within the synaptic knob. So these vesicles, they'll fuse with the presynaptic membrane and they release neurotransmitter by the process of exocytosis. The synaptic vesicles release the neurotransmitter through a small hole in the cell membrane, and then this opening will reseal rapidly and the main vesicle stays inside the cell, and this process is called kiss and run discharge. I'll show you in the next slide about this picture. So about this presynaptic membrane, it is just an axonal membrane, and there are some thickened areas in the presynaptic membrane which are called as active zones, and only in these active zones the synaptic vesicles comes and fuse and release their contents into synaptic cleft. So near the active zones there will be voltage gated calcium channels will be present. So we should remember 2 points about this presynaptic membrane. It is the axonal membrane which is lining the nerve terminal, and it is the one which contains mainly the voltage gated calcium channels, and the thickened region of this presynaptic membrane is called active zones. It is the place where the synaptic vesicles will come and fuse with the fuse, and it will discharge its contents into the synaptic cleft. So the next is synaptic cleft. Synaptic cleft is the space between the presynaptic membrane and the postsynaptic membrane, which is actually 20 to 40 nanometer wide space, which is filled with the extracellular fluid with reticular fibers forming the matrix. And this is the postsynaptic membrane. So it is the muscle fiber membrane. Simply we can say it is a sarcolemma. The muscle fiber membrane, you can see it is thickened and depressed to form synaptic trough, which is also called as junctional folds. So what is this junctional folds? This postsynaptic membrane or the muscle fiber membrane is thickened and depressed to form the synaptic trough, and that is called as junctional folds. This is the place where this terminal buttons will fit into the postsynaptic membrane. So there is some thickened portion of the muscle fiber membrane which is called as end plate. So on each end plate only one nerve fiber will end. There is no convergence of or multiple inputs. So just remember, on each end plate only one nerve terminal will fit. So there is no convergence. Postsynaptic membrane is thrown into large number of folds which are called as subneural clefts, and the matrix of the subneural cleft contain enzyme which is called as acetylcholine esterase, and it is a very important enzyme because it is the one which helps in the degradation of the acetylcholine into acetate and choline. So all these are the structures. So we will see many more pictures. So I told you, so here we can see here you can see this is the presynaptic membrane where it contains the synaptic vesicles. They are going to bind with the presynaptic membrane and they are going to release their contents into synaptic cleft, right? So here the neurotransmitter is released into the synaptic cleft. And very important point we missed is the postsynaptic membrane contains the acetylcholine receptors. So they are called as nicotinic acetylcholine receptors, which is also called as acetylcholine gated ion channels. So they contain postsynaptic membrane contains the nicotinic acetylcholine receptors, which is also called as acetylcholine gated ion channels. I'll show you how the structure will be. So here you can see the axon, it loses its myelin sheath and it divides into a number of branches and it is going to innervate the skeletal muscle fiber. So that is the junction which is called as neuromuscular junction. Here you can see the synaptic knob, the presynaptic membrane, there is a space which is a 20 to 40 nanometer space which is synaptic cleft, and then the postsynaptic membrane, it is thrown into folds which is called as junctional folds, and the thickened region is called as subneural clefts, and in this matrix of the subneural clefts we have this acetylcholine esterase enzyme, and the postsynaptic membrane contains the nicotinic acetylcholine receptors. So the presynaptic membrane contains voltage gated calcium channels very close to the active zones. So active zone is the place where the synaptic vesicles will come and fuse and it will release its contents into the synaptic cleft. So here you can see very clearly that the presynaptic membrane is thickened some region, that is the zone which is called as active zone, and in this place only the synaptic vesicles are coming and fuse and they're going to release their contents. So here you can see the presynaptic membrane contains voltage gated calcium channels, and postsynaptic membrane contains acetylcholine receptors. So how the neuromuscular transmission occurs? So when the action potential reaches the nerve terminal of the motor neuron, that is the nerve terminal, what happens? There is opening of the voltage gated calcium channels. So we said that presynaptic membrane contains voltage gated calcium channels. Whenever the action potential reaches to the nerve terminal, the voltage gated calcium channels will open up. This increases calcium influx from the extracellular fluid which is filled in the synaptic cleft. From there, the calcium will enter inside the cell, and within the nerve terminal there will be increase in the calcium level. So this increase in the calcium level will cause fusion of the synaptic vesicles with the presynaptic membrane. We told that in the active zones, the synaptic vesicles will come and fuse. I already told you what is kiss and run discharge. That is, the synaptic vesicles will come and fuse with the presynaptic membrane, and there will be a small hole which is formed in the membrane of the the synaptic vesicle, which will release their contents into synaptic cleft, and then they reseal and they move rapidly into the nerve terminal, and this is called kiss and run discharge. So the release of the mechanism by which the neurotransmitter is released into the synaptic cleft is by the process of exocytosis. So what happens when an action potential reaches the nerve terminal? Voltage gated calcium channels open, and there is increase in the intracellular calcium level, which causes fusion of the synaptic vesicles with the presynaptic membrane. By the process of exocytosis, the acetylcholine is released into the synaptic cleft. This acetylcholine diffuses and reaches the postsynaptic membrane, where it binds with the acetylcholine receptors. So when the acetylcholine molecules are binds with the acetylcholine receptors, this allows increase in the sodium influx into the muscle fiber membrane. So within the muscle fiber membrane, there is because of sodium influx, depolarization occurs. This depolarization is just it can create a local potential that is called as end plate potential. So the local end plate potential is developed, which is then can be summated, and it can produce action potential when it reaches when this end plate potential reaches a threshold level of -16 millivolts. It produces an action potential. And there is a term which is called as miniature end plate potential. What happens when even at rest, a small amount of acetylcholine is released, and that can produce a 0.5 millivolt depolarization of potential, that is called miniature end plate potential. And this acetylcholine which is released will be degraded by the acetylcholinesterase enzyme. I already told you the matrix of the subneural clefts contain the acetylcholinesterase enzyme, which helps in the degradation of the acetylcholine into acetate and choline. So these are the steps in the neuromuscular transmission. So first what happens? The action potential reaches the nerve terminal. Next what happens? Voltage gated calcium channels open and calcium influx occurs. So there is increase in the intracellular calcium within the nerve terminal. There is increase in the calcium concentration. This causes fusion of the synaptic vesicles with the membrane, and there are some proteins which helps in the fusion of this synaptic vesicles with the membrane. Those proteins are called as docking proteins. So next what happens? I'll show you that picture also. So next what happens? This synaptic vesicles, they'll release their contents into synaptic cleft, and they diffuse and they binds with the acetylcholine receptors which is present in the postsynaptic membrane. This allows, when there is opening of this gates in the acetylcholine channels, the sodium influx will occur, and this produces end plate potential, which is then summated and forms the action potential, which can be propagated. And this action potential spreads on either sides of the muscle fiber membrane, that can produce muscle contraction. So how the neurotransmitter is released? So here you can see actually from the early endosome, the synaptic vesicle will bud off, and then it is filled with neurotransmitter. I told you neurotransmitter and synaptic vesicle both are synthesized from the cell body of the spinal motor neurons. So the neurotransmitter which is synthesized from the cell body, it is filled in the synaptic vesicles, which is then translocated to the presynaptic membrane. In the presynaptic membrane, it is docked and primed, and this is the docking occurs by the proteins which are called as docking proteins. There are two proteins which are called as synaptobrevin and syntaxin. Synaptobrevin is a docking protein which is present in the membrane of the synaptic vesicle, whereas syntaxin is a membrane which is present in the presynaptic membrane. So these two get locked and this docking occurs. So those are called as docking proteins, which is then primed. And I told you, whenever there is action potential reaches, whenever there is opening of the voltage gated calcium channels, this allows increase in the calcium level. So next what happens, this synaptic vesicle will come and fuse, and by the process of exocytosis they'll release their contents, and this empty synaptic vesicles they'll again reseal and they'll be translocated again by the process of endocytosis, which is actually a clathrin mediated endocytosis. You can see these proteins are called as clathrin proteins, which will help in the which will mediate the process of endocytosis. So this is how the neurotransmitter will be released and the vesicles will be recycled. So here you can see a picture which is showing the docking proteins. So here you can see synaptobrevin. I told you it is a docking protein which is present in the membrane of the synaptic vesicle, and here we have in the presynaptic membrane we have syntaxin, and these two proteins lock and the docking mechanism occurs. So synaptobrevin is the T-SNAP protein, whereas syntaxin is a V-SNAP protein. So these two proteins helps in the docking of the synaptic vesicle with the membrane, presynaptic membrane. So here you can see a structure of the acetylcholine receptor. So we told postsynaptic membrane contains acetylcholine receptor, which is also called as acetylcholine gated ion channel. It is a nicotinic acetylcholine receptors. So it is a protein structure which is having 5 subunits, 2 alpha subunits, beta, gamma and delta subunits, and with these 2 alpha subunits, 2 molecules of acetylcholine will bind, and that will open the this gated ion channel that will allow the sodium influx. And why there is sodium influx when there is opening of the acetylcholine gated ion channel? Because this acetylcholine gated ion channel is the inside of the acetylcholine gated ion channel will be negatively charged membrane, and because of this the negatively charged particles will be repelled and only positive ions will be rushed in through this acetylcholine receptors. You know that sodium is the major cation in the extracellular fluid, and that is why sodium influx mainly occurs. Even sodium, magnesium and even the calcium can also pass through this. Sodium, magnesium and potassium can also pass through this acetylcholine gated ion channels, but sodium, because concentration gradient is so much, so the sodium influx occurs, which is helpful in the depolarization of the muscle fiber membrane. So here you can see it is a acetylcholine gated ion channel. So here we have the acetylcholine receptors. Here you can see the gates are closed, and when the acetylcholine molecules comes and binds with these acetylcholine receptor, these gates open and sodium influx occurs through this acetylcholine gated ion channels. So here this is the picture to summarize the steps in the neuromuscular junction. So when the action potential reaches the nerve terminal, there is opening of the voltage gated calcium channels which is present in the presynaptic membrane, that increases the calcium influx, and that allows the fusion of the synaptic vesicles with the presynaptic membrane. By the process of exocytosis, acetylcholine is released into the synaptic cleft, which is then diffused and reaches the postsynaptic membrane, where it binds with the nicotinic acetylcholine receptors, which allows the influx of sodium, which is then producing end plate potential, which is a local potential. It can be summated where it cannot be propagated. It does not obey all or none law. It cannot be propagated, and this end plate potential, when it reaches a threshold level of minus 60 millivolts, that produces an action potential, and this action potential spreads on the muscle fiber membrane, which is helpful in the muscle contraction. So here we can see the potentials. I told you, so A is the end plate potential and B is the action potential, C is the miniature end plate potential. So here you can see actually the resting membrane potential of skeletal muscle is minus 80 to minus 90 millivolts. So here you can see minus 90 millivolts, that is the resting membrane potential of skeletal muscle. So from resting membrane potential, when depolarization occurs, when the sodium influx occurs, what happens? End plate potential is developed. But when it reaches threshold level of minus 60 millivolts, it is producing an action potential. And even at rest small amount, that is 0.5 millivolts of depolarization occurs, that is the potential which is called as miniature end plate potential. So this acetylcholine, how it is synthesized and how it is removed. This acetylcholine, there are within the synaptic, there are 10,000 molecules of acetylcholine within each synaptic vesicles, and at one time, whenever there is exocytosis, 125 vesicles burst by each exocytosis. So I told you there are 3 lakhs synaptic vesicles which are present in the synaptic knob or nerve terminal. So here you can see acetyl-CoA and choline both are combining to form acetylcholine molecules. So this acetylcholine, it is degraded by the enzyme acetylcholine esterase, which is present in the matrix of the subneural cleft. It is degraded into acetyl-CoA, acetate and choline, and the choline is a precious material, so it is taken into the synaptic cleft and it is reused for the synthesis of acetylcholine. So this is the function of acetylcholinesterase enzyme. So try to answer for this MCQs. What is the function of synapsin? So I told you synapsin and synaptobrevin both are the docking proteins, right? They help in the fusion of the synaptic vesicles with the presynaptic membrane. So try to answer for this question. It activates calcium colmogulin dependent protein kinase. It's an anchor protein of acetylcholine vesicles to the presynaptic terminal. Helps to open voltage gated calcium channels in presynaptic membrane. Substances necessary for neurotransmitter synthesis. So answer is B, that is it is the anchor protein of acetylcholine vesicles to the presynaptic terminal. A potential developed at the post-junctional membrane in a neuromuscular junction is receptor potential, pacemaker potential, end-plate potential, action potential. So it is end-plate potential. I told you the postsynaptic membrane is also known as end-plate. So the potential which is developed in this postsynaptic membrane is called end-plate potential, which is a local potential, which is a graded potential, but it can be summated and can produce action potential. End-plate potential has the following characteristics except it is graded, it is non-propagated, it has no latency, it is propagated. So which is the exception? So it is, yes, it is a graded potential. It cannot be propagated, it has no latency, it is propagated. So only action potential can be propagated. So answer is D. Neurotransmitter is released at the neuromuscular junction due to, so you know this, right? So whenever there is calcium or voltage gated calcium channels opens, influx of calcium occurs, that will cause the fusion of synaptic vesicles, which will allow the release of neurotransmitter. So entry of calcium ion is the answer. Which one of the following is the site for the neurotransmitter synthesis? Dendrite, nerve cell body, axoplasm, terminal patterns. Which is the site of synthesis of neurotransmitter? Yes, it is the site. It is the nerve cell body, which is the cell body of the motor neurons, is the site where the neurotransmitter is synthesized, because that is the one which contains the protein machinery, that is the enzymes and proteins are synthesized from the nerve cell body. So even neurotransmitter is synthesized from the nerve cell body. So it is then transported to the nerve terminal by the fast axoplasmic transport, that is the acetylcholine molecules. Spontaneous release of acetylcholine at the neuromuscular junction produces, so miniature end-plate potential, action potential, post-tetanic potential, resting membrane potential. So spontaneous release, I told you, even at rest there is small amount of acetylcholine that is released, and that can produce action, that can produce a potential which is called as miniature end-plate potential. So the answer is CA, that is